Namaste to all students. Khushbu Vidya, myself JV Dr. Khushbu Verma from Faculty of Agriculture and Veterinary Science. Today we are going to discuss about Resection endonuclease enzyme. This session is belong to recombinant DNA technology course of program BTech FPT 4th semester. So first of all, you have to know about the resection endonucleases and what is the role of resection endonuclease enzyme in recombinant DNA technology. So first of all, prokaryotic nucleases have been isolated and have been identified that recognize a specific DNA sequence and cleave sugar phosphate backbone, either within or close to recognition site. These enzymes are produced by bacteria and archaea for defending themselves against the foreign DNA molecules. So that was the reason to produce resection enzymes in prokaryotic cells. So, and resection enzymes recognize specific sequences and make cuts in the DNA molecules. And Stuart Lin and Werner Arber in 1968 showed in vitro resection of FD phage DNA by an E. coli cell extract. Means it was the first demonstration regarding resection enzymes done by Stuart Lin and Werner Arber in 1968. Soon after this discovery, Hamilton Smith and Kent Wilcox in year 1970 isolated the first resection enzyme, which was endonuclease R. Later, it was renamed as Hind second. From Hind second, this name for the resection enzyme has been given because it has been isolated from Haemophilus influenzae, and it was the second strain which was identified. That's why we call it Hind second. And in the subsequent year, Kathleen Dana and Daniel Nathans pioneered the applications of the resection enzymes by showing specific cleavage of SV40 DNA by Hindisecond. Further, Werner Arber and Hamilton Smith and Daniel Nathans were awarded for Nobel Prize in Physiology or Medicine in 1978 for the discovery and the applications of the resection enzymes, because it was a huge discovery, because now you can cut and trim the target DNA by using naturally produced resection enzymes. So after the isolation of Hind second in 1970s, many other resection enzymes also have been isolated from several bacterial and archaeal sources. Comprehensive information about all the characterized resection enzymes is available in the database known as Rebase, given by R.J. Roberts and his colleagues in 2007. As per this database, 3,805 resection enzymes have been biochemically or genetically characterized till now. Resection enzymes have been classified into four major types known as type 1, type 2, type 3, and type 4, basically depending on their composition, cofactor requirement, target sequences, position of cleavage site, etc. Among these, type 2 enzyme have separate enzymes for the endonuclease and methylase activities. A type 2 enzyme basically cuts at or near its recognition site, so hence type 2 enzymes have been found to be most useful in recombinant DNA technology. On the other hand, type 4 recognized modified, typically methylated DNA only. As per the Rebase database, 611 type 2 resection enzymes are commercially available till now. Werner Arber in 1929, June 3rd, he basically Nobel Prize in Physiology of Medicine in 1978, and basically he showed in vitro resection of FD phase DNA by E. coli extract. This is the major history related to the resection endonuclease enzyme second one was the Hamilton O. Smith. He was also awarded by the Nobel Prize in 1978, and he isolated and characterized the first resection enzyme, endonuclease R, which was given as a renamed in the second. Next one was the Daniel Nathans and Kathleen Janet Dana. Both pioneered the application of resection enzymes, which is basically specific cleavage of SV40DNA. If we talk about the resection enzymes type, and so in type 1 example is EcoK and EcoP. Type second example is EcoR1 and EcoR5. Type third example is EcoP1 and EcoP15. Type 4 example is MCR, BC and MRR. So these are the examples of each type of resection endonuclease enzyme. On cutting of the DNA at specific sequences, some of the type second enzymes produces blunt ends. The cut sites for type second enzyme like SMA1 have been shown in the below, and you can understand this cutting pattern by the diagrammatic representation, which I am showing here in the presentation. Here you can simply see this is SMA1, and it has the activity to create blunt end only. It will cut in between the CCC GGG portion at the both side. So they will create the blunt end, means they are not having any flanking regions or unpaired region, which is also known as sticky ends. And some of the other type second enzymes produces cohesive ends or sticky ends. So the cohesive ends may have 5' overhang, as has been shown below in the case of BAM-H1 enzyme. BAM-H1 enzyme basically cut in between the G and G, here G and G. So it will create like sticky ends at the 5' end. And in the other example, cohesive ends produced by some of the type second resection enzymes have 3' overhangs as given below in case of PST1 enzyme. So PST1 enzyme will create the cut in between A and G. It will create like this and then this, and that's how they will have 3' and overhang. Now we can discuss about the comparative study of each type of resection enzyme. So you can simply see this type 1, type second and type third, and there are the properties. Like if we discuss the comparison in between all these three types, so first property is enzyme activities and structure. So in type 1, this is basically single multi-subunit having both endonuclease and methylase function. They are large in the size. And type second has separate endonuclease and methylase enzymes, and they are small in the size. On the other hand, type third basically have separate subunits for restriction and methylation. And there is no evidence about the size or large size or small size. And second property is basically recognition sequence. So type 1 basically asymmetric and bipartite, and on the other hand, type second is palindromic with dyed symmetry, and type third basically has recognition sequence for as asymmetric unipartite. Third property is recognition site length. So type 1 will recognize the long length of 8 to 16 base pair. Type second will recognize short length of 4 to 9 base pair. Type third will also recognize the short length of 5 to 7 base pair. Fourth property is methylation site. So type 1 has the methylation site similar to the recognition site. Type second also recognition site, and type third also recognition site, where they do the methylation process. Next property is requirement for the resection. So type 1 require ATP for the energy generation because they will hydrolyze the ATP and convert into ADP, and after the hydrolysis process energy will generate, and type 1 use this energy for the resection process and Mg plus 2 and SEM. And type second is having only requirement of Mg plus 2, one is there is no need of energy consumption. Type third also have use of ATP and Mg plus 2. SEM is only optional. Next property is cleavage site. So type 1 cleavage site is not specific, it is basically random, and it will recognize the site 1000 base pair away from the actual site of the recognition. Type second, approximately at the recognition site, they will have the cleavage site or just near about the recognition site. Type third is having approximately 25 base pair downstream of the recognition site. Examples of type 1 is eco K, eco B. For type 2 is eco R1, eco R5, and for type 3 is eco P1 and eco P50. Now we have to discuss about the types of resection sites. So first one is palindromic recognition sequences. What is the meaning of palindromic recognition sequence? So sequence having two-fold axis of dyad symmetry. That is, phrases that read the same backwards as forward. Means, madam, I am Adam. Continuous means two half sides of the recognition sequences are adjacent. Like, for example, 5 and GAATTC. Third, and discontinuous means two half sides are separated or interrupted. Next one is the uni or bipartite recognition sequences. Means an interrupted non-palindromic recognition sequence are known as uni or bipartite recognition sequences. You can simply see here about the difference or about the specificity of these kind of a sequence. You can simply see this is ATG, CC, multiple N, and then TAGCG. So this is the uni or bipartite sequence. On the other hand, this is palindromic sequence. Madam, I am Adam. Means MADAM, MADAM, MADM, and MADM. So this is the basically difference in between palindromic sequences and uni-bipartite recognition sequences. Now come to the classification on the basis of length of recognition. So sequences or fragment size. So frequency of occurrence of any recognition sequence in random sequences of DNA is given by 1 by 4n, where n is length in base pair of recognition sequence. For example, a 4-base cutter that recognizes a tetranucleotide recognition sequence would cleave DNA every 44 is equal to 256 base pair. Classification of bases of types of and generated blunt end cutters. So endonucleases cut straight across the axis of symmetry of recognition site, means they will not create any kind of a sticky. On the other hand, if enzymes are sticky and cutters, so endonucleases enzymes which cuts the DNA of the center of resection site, but between two same bases on opposite direction. Here you can see the diagrammatic example of sticky and cutter endonuclease enzyme. You can simply see this is 5 dash overhang cutting, sticking and generation by eco-R1. So eco-R1 basically recognize GA region and it creates this kind of a zigzag cutting, and basically it produced the 5 dash end. So 5, this is the 5 dash end and this is the 5 dash. In the third dash overhang, PST will cut this end and they will create third dash overhangs. So this is how resection enzymes and resection endonuclease enzymes do their work. And there are some many key points or many important points for competitive exams. Like first of all, types of are DNA endonuclease enzyme, resection endonuclease enzyme, and which resection endonuclease enzyme type do not require any kind of a ATP hydrolysis, or they are not consuming energy consuming enzymes. So this is the major question. And one more thing, which resection enzyme has same or sharing methylated or endonuclease activity both. So this is also a major point. And one more thing, what is the difference in between a plantain and sticky ends, and what are the 5 dash overhang sticky end and third dash overhang sticky ends. And also you have to learn about the history of the restriction enzyme, and like which was the first restriction enzyme which was first isolated, and who gave the in vitro restriction demo at first, and who like the scientists were awarded with the Nobel Prize in the Physiology or Medicine in 1978 for the discovery and application of the resection enzymes. So all these are the key points for important for the competitive exam prospectus. So this is the sum up of this session, and this session is powered by digital version 2.0 JAYOTI VIDYAPEETH Women's University Jaipur. If you have any query, then mention in the comment box. I will resolve it. Thank you.